Good evening, dear students. Welcome to Sankal. So, in this session, we are going to learn about biotechnology principle and processes. In the last session, we learned about 1 mark question or MCQs, what we say, right? So, here, yeah. So 1 mark question, MCQs. So we solved around 25 PU board based MCQs, multiple choice questions in the last session. So according to the blueprint, what you see here is total marks 7 we have, out of which 2 we will get MCQs, right? So one question we will get for 5 marks, that is long answer question. Okay, and be prepared that this 5 marks question will not be so direct question. I mean to say, so easy question. Directly they will not ask define, describe, explain. So it will be a high order thinking question, or what we call it as HOTS, right? So let us proceed with the same kind of questions. So we have around 6 to 7 questions which are 5 marks questions, which we are going to discuss today. So in this, what we are going to do, I will be explaining the question, right? I will be telling the question. Then after that, you have to recall the answer. What answer you can write for this question? And I will tell you what answer and how to write the answer and which words are compulsory for that question. Which words you have to use, which scientific words you have to use in that question, which I am going to tell you. Okay, so let us begin without further delay. Yeah, okay, just a minute. So we have 5 marks questions here. Here it is. Fine. So 5 marks question. The first question goes like this. Write 3 vector-free techniques that can be used to transfer recombinant DNA into a ready host cell. Okay. Then in this only we have another question. Agrobacterium tumefaciens acts as a natural vector for cloning genes in plants. Substantiate. So when you see these questions, you will understand that we have 3 marks question here and 2 marks question here. Okay. So as we have 3 marks question here, okay, here we have to write around 6 points, what we can say, 5 to 6 points. Then which we have to write, which points we have to write, we are going to discuss. Next 2 marks question, around 3 to 4 points. Just keep it in your mind. 5 marks, actually 10 points has to be dealt. So as they divided 5 marks into 3 marks and 2 marks, we have to also divide the points. So write the vector-free techniques that can be utilized to transfer recombinant DNA into a ready host cell. In some of the experiments, right, so we cannot use the vector. Okay, we cannot use the vector. So instead of vector, we are using certain tools and techniques to transfer the recombinant DNA. Which are those techniques that you have to write? And you have to write 2 sentences regarding that technique. So let us see what will be the answer. Okay, the next slide is, yes, answer. Okay, yeah. See here, the first point in vector-based free method is electroporation, what we call. So like this, you have to write the subheading electroporation, because your representation of what we can say, the answer sheet is also necessary. So electroporation means what? Applying an electric field to the cells. So when you apply the electric field, what happens? It increases the permeability of cell membrane. So we know that the cell membrane is a semi-permeable membrane. It will not allow all the materials to get inside it. It will not allow all the materials to go outside it. So to increase the permeability of the cell membrane, we are going to apply the electric field. So when you apply the electric field, that allows the recombinant DNA to enter the host cell. So this is one method what you say. Electroporation. After electroporation, micro injection we will use. So what is this technique? It is a technique where a fine needle is used directly inject the recombinant DNA into the nucleus of the host cell. So when you see, yeah, so when you see here, which are the key terms? Fine needle, right? So directly into the nucleus of a host cell. This is required. This method is particularly effective to transferring the DNA into animal cells. So microinjection is used to transfer the rDNA into the animal cell, what we call, right? Then after that, chemical transformation. So I said 3 points should be included, that it has three methods which are vector free. So the next one is chemical transformation. Chemical transformation uses what? As the name suggests, it uses chemical solutions, my dear friends. So it uses chemical solutions like calcium chloride to make the cell membrane more permeable. When the cell membrane becomes more permeable, the recombinant DNA is then added and heat shock is applied to facilitate uptake by the host cells, what we call. So these are the vector free methods which we use in the genetic engineering. So this is for what? This is for our first question, vector free based methods. So 3 points are there. Half marks for your subheading, half marks for your description. Like this you have to write the answer. Okay na? Next 2 marks question, agrobacterium ಬಗ್ಗೆ ಇದೆ. Yeah. ಸೊ ಅಗ್ರೋಬ್ಯಾಕ್ಟೀರಿಯಮ್ ನ್ಯಾಚುರಲ್ ವೆಕ್ಟರ್ ಆಗಿ ಆಕ್ಟ್ ಆಗತ್ತೆ. ಯಾವ ರೀತಿ ಆಕ್ಟ್ ಆಗತ್ತೆ ಅನ್ನೋದು ಕ್ವಶನ್ ಇದೆ. ಸೊ ಸಿ ಹಿಯರ್ ವಾಟ್ ಯು ಹ್ಯಾವ್ ಟು ರೈಟ್. ಸೊ ಲೆಟ್ ಮಿ ಟೆಲ್ ಯು, ನಾನು ಹೇಳ್ದೆ ಪಾಯಿಂಟ್ ವೈಸ್ ಬರಿಬೇಕು ಅಂತ, ಇಲ್ಲಿ ಪ್ಯಾರಾಗ್ರಾಫ್ ವೈಸ್ ಇದೆ. ಸೊ ವೆನ್ ಯು ಜಸ್ಟ್ ಕಟ್ ದ ಪ್ಯಾರಾಗ್ರಾಫ್ ಯು ವಿಲ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್. Agrobacterium tumefaciens, actually it should be in italics as yeah, it should be in italics or you should underline it. Okay, acts as a natural vector. ಯಾಕೆ ಅಂದ್ರೆ it has a specialized plasmid which is called as T plasmid. ಇದೇನ್ ಮಾಡತ್ತೆ? For cloning genes in plants by transferring its tDNA into the plant genome, right? So utilizing its T plasmid which can be modified to carry desired gene. So T plasmid ಯಾವ ರೀತಿ ಇದೆ ಅಂದ್ರೆ ಅದನ್ನ ನಾವು modify ಮಾಡ್ಬೋದು. ನಮಗೆ ಯಾವ ಜೀನು ಅದು ಕ್ಯಾರಿ ಮಾಡಬೇಕು ಅಂತ ಅದಕ್ಕೆ ತಕ್ಕ ಹಾಗೆ ಆ ಟಿ ಪ್ಲಾಸ್ಮಿಡ್ ಅನ್ನ ಮಾಡಿಫೈ ಮಾಡಬಹುದು. ಸೊ ಅದು ಇದು ಅಗ್ರೋಬ್ಯಾಕ್ಟೀರಿಯಂ ಟ್ಯುಮಿಫೆಸಿನ್ಸ್ ಅನ್ನೋ ಬ್ಯಾಕ್ಟೀರಿಯಾದ ಸ್ಪೆಷಾಲಿಟಿ. ಸೊ ದಟ್ ಈಸ್ ವೈ ದಿಸ್ ವೆಕ್ಟರ್ ಈಸ್ ಯೂಸ್ಡ್ ಅಂಡ್ ಇಟ್ ಆಕ್ಟ್ಸ್ ಆಸ್ ಅ ನ್ಯಾಚುರಲ್ ವೆಕ್ಟರ್ ಆಸ್ ಇಟ್ ಹ್ಯಾಸ್ ಟಿ ಪ್ಲಾಸ್ಮಿಡ್. ಸೊ ಇವೆಲ್ಲವೂ ಕೂಡ ನಿಮ್ದೇನಾಗತ್ತೆ ಕೀವರ್ಡ್ಸ್ ಆಗ್ತವೆ. ಓಕೆನಾ? ಸೊ ದಿಸ್ ಇಸ್ ರಿಗಾರ್ಡಿಂಗ್ ದ ಫಸ್ಟ್ ಕ್ವಶನ್ ವಾಟ್ ವಿ ಸೇ. ದ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ಇಸ್ ಯಾ. ಸೊ ದಿಸ್ ಕೈಂಡ್ ಆಫ್ ಕ್ವಶನ್ ಇಸ್ ಮೋಸ್ಟ್ ಒಂದ್ಸಲ ಆದ್ರೂ 5 ಮಾರ್ಕ್ಸ್ ಕ್ವಶನ್ ಅಲ್ಲಿ ಈ ರೀತಿ ಕ್ವಶನ್ ಇರುತ್ತೆ. ಸೊ ಒಂದು ಟಾಪಿಕ್ ಅನ್ನ ಕನ್ಸಿಡರ್ ಮಾಡ್ತಾರೆ, ಆ ಟಾಪಿಕ್ ಅಲ್ಲಿರುವ ಮೇನ್ ಏನು ಅಪ್ಲೈಡ್ ಕ್ವಶನ್ಸ್ ಗಳಿರ್ತಾವೋ ಅದರ ಬಗ್ಗೆ ಗಿವ್ ರೀಸನ್ಸ್ ಅನ್ನ ಕೊಡಿ ಅಂತ ಹೇಳ್ತಾರೆ. ಸೊ ಗಿವ್ ರೀಸನ್ ಫಾರ್ ದ ಫಾಲೋಯಿಂಗ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಅಂತ ಇದೆ. ಫಸ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಏನಿದೆ? Alien DNA is linked with ORI site of a vector in gene cloning. ಯಾಕೆ ORI site ಗೆ ಯಾಕೆ ಅದನ್ನ ಲಿಂಕ್ ಮಾಡ್ತಾರೆ? Why not to other site? That you have to give the reason. B is restriction enzymes are called molecular seizures. ಯಾಕೆ ನಾವು RENs ಅನ್ನ molecular seizures ಅಂತ ಕರಿತೇವೆ? C, DNA ligase can be called molecular glue or molecular stitcher. ಯಾಕೆ? Gel electrophoresis is considered as a very important technique in recombinant DNA technology. DNA fragments move towards anode under electric field through a medium in gel electrophoresis. So for all these statements, you have to give 2 to 3 sentences of reason. And the keywords are necessary and you should underline the keywords. So let us see the answer here. The first one. What is the first question? Alien DNA is linked with oricite. Yake? So to enable the alien DNA to replicate and multiply itself in the host organism. So alien DNA ಅಥವಾ foreign DNA ಅಂತ ಏನು ಹೇಳ್ತೇವೆ which carries our desired gene, it has to be multiplied, alva? It has to replicate. That is why we are going to attach it at the oricite. ಯಾಕೆ ಅಂದ್ರೆ oricite ಅಲ್ಲಿ ಏನಾಗತ್ತೆ replication start ಆಗತ್ತೆ. Okay, so this is about the first question. Next we have is restriction enzymes ಅನ್ನ molecular seizers ಅಂತ ಕರಿತೀವಿ. ಯಾಕೆ? So restriction enzymes are called as molecular seizers because they cut the DNA at specific sequences. At specific sequences. Can you recall at which sequences? ಯಾವ specific sequence ಅಲ್ಲಿ cut ಮಾಡ್ತವೆ ಅವು? What are those sequences are called as? We studied in multiple choice questions ಅಲ್ವಾ? Palindromic sequence ಅನ್ನ ಹುಡುಕಿ palindromic sequence ಹತ್ರ ಏನ್ ಮಾಡ್ತವೆ? DNA ಅನ್ನ cut ಮಾಡ್ತವೆ. So that is why these are called as what? Molecular seizers. Sudden ಆಗಿ ಸೀಸರ್ ತರ ಎಲ್ಲಿ ಬೇಕಾದ್ರೂ ಕಟ್ ಮಾಡಲ್ಲ ಇದು. At a specific site ನಮ್ಮ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಎಲ್ಲಿರುತ್ತೆ ಅಲ್ಲೇ ಹೋಗಿ ಅದೇನ್ ಮಾಡುತ್ತೆ ಪ್ಯಾಲಿಂಡ್ರೋಮಿಕ್ ಸೀಕ್ವೆನ್ಸ್ ಅನ್ನ ಕಟ್ ಮಾಡುತ್ತೆ. That is why this restriction endonuclease enzymes are called as molecular seizers. Okay. Next. Yeah. DNA ligase ಗೆ ನಾವು molecular glue ಅಂತ ಕರಿತೇವೆ. ಯಾಕೆ? So it joins two DNA fragments. DNA fragments ಅಲ್ಲಿರುವ sticky ends ಅನ್ನ ಏನ್ ಮಾಡುತ್ತೆ ಇದು join ಮಾಡ್ತವೆ. By forming what? By forming phosphodiester bond between the 3' OH end of one fragment, 5' end of the other. So ಒಂದು fragment ನ 3' ಎಂಡು ಇನ್ನೊಂದು fragment ನ 5' ಎಂಡು ಅಟ್ಯಾಚ್ ಆಗ್ಬೇಕು. ಹೌದಾ? So that will be done by what? DNA ligase. So that is why these are called as molecular seizers. Okay. Next. Yeah. So gel electrophoresis. So what is the question there? Just a minute. I will just read the question. Gel electrophoresis is considered as very important technique. ಯಾಕೆ ಅದನ್ನ ನಾವು ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಟೆಕ್ನಿಕ್ ಅಂತ ಕನ್ಸಿಡರ್ ಮಾಡ್ತೀವಿ ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗ್ ಅಲ್ಲಿ? ಯಾಕೆಂದ್ರೆ ದಿಸ್ ಮೆಥಡ್ ಈಸ್ ಯೂಸ್ಡ್ ಟು ಸಪರೇಟ್ ದ DNA ಫ್ರಾಗ್ಮೆಂಟ್ಸ್. ಯು ಕ್ಯಾನ್ ಸಿ ದ ಡೈಗ್ರಾಮ್ ಇನ್ ಯುವರ್ ಟೆಕ್ಸ್ಟ್ ಬುಕ್ ಅಲ್ವಾ? ಸೊ ಜೆಲ್ ಎಲೆಕ್ಟ್ರೋಫೋರೋಸಿಸ್ ಅಲ್ಲಿ DNA ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಬೇಸ್ಡ್ ಆನ್ ದೇರ್ ಸೈಜ್ ಏನಾಗ್ತವೆ ಸಪರೇಟ್ ಆಗ್ತವೆ. ದಿಸ್ ಈಸ್ ಇಂಪಾರ್ಟೆಂಟ್ ಬಿಕಾಸ್ it allows you to check if a PCR reaction worked or if a plasmid has a correct gene. ನಾವು ತಗೊಂಡಿರೋ ಪ್ಲಾಸ್ಮಿಡ್ ಅಲ್ಲಿ ರೀಕಾಂಬಿನೆಂಟ್ DNA ಆಗಿದೆನಾ, ನಮ್ಮ ಜೀನ್ ಆಫ್ ಇಂಟ್ರೆಸ್ಟ್ ಇದೆನಾ ಅನ್ನೋದನ್ನ ಚೆಕ್ ಮಾಡೋದಕ್ಕೆ ದಿಸ್ ಎಲೆಕ್ಟ್ರೋಫೋರೋಸಿಸ್ ಏನ್ ಮಾಡಬೇಕು? DNA ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಅನ್ನ ಸಪರೇಟ್ ಮಾಡಬೇಕು. ಸಪರೇಟ್ ಮಾಡಿದ ಮೇಲೆ ಏನಾಗತ್ತೆ PCR ರಿಯಾಕ್ಷನ್ ಅಲ್ಲಿ ನಮಗೆ ಈ ಒಂದು ಪ್ಲಾಸ್ಮಿಡ್ ಕರೆಕ್ಟ್ ಜೀನ್ ಅನ್ನ ಹೊಂದಿದೆ ಇಲ್ವೋ, whether it has a correct gene or not, that we will understand by the gel electrophoresis what we say. Right. The next one is, they asked DNA ಅನ್ನೋದೇನಾಗತ್ತೆ, these DNA fragments in the gel electrophoresis what we call, so these will move towards anode. Very easy question actually. If you understood this chapter ಅಲ್ವಾ ನಿಮಗೆ ಈಸಿಯಾಗಿ ಅರ್ಥ ಆಗತ್ತೆ. ಸೊ ನಮಗೆ ಗೊತ್ತಿರೋ ಹಾಗೆ DNA is made up of what? Sugar and phosphate backbone. ಹೌದಾ? ಸೊ ಫಾಸ್ಫೇಟ್ ಅನ್ನ ನೀವು ಬರೆದ್ರೆ PO4 3-, ರೈಟ್? ಸೊ ಇದು ಏನು? ದಿಸ್ ಈಸ್ ಅ ನೆಗೆಟಿವ್ಲಿ ಚಾರ್ಜ್ಡ್ ಅಯಾನ್. ಯಾವುದು? ಈ ಫಾಸ್ಫೇಟ್. ಸೊ ಯಾವಾಗ ನಾವು ಎಲೆಕ್ಟ್ರಿಕ್ ಕರೆಂಟ್ ಅಪ್ಲೈ ಮಾಡ್ತೀವೋ, ದಟ್ ಈಸ್ ವೆನ್ ಯು ಅಪ್ಲೈ ದ ಎಲೆಕ್ಟ್ರಿಕ್ ಕರೆಂಟ್ ಟು ದ ಜೆಲ್, obviously this negatively charged particle move towards its opposite pole. Which is its opposite pole? It is nothing but a anode, which is a positive side, which is positively charged electrode. Okay na? So this is the reason for this question. So I hope you understood. See, for every statement you have to give the reason. You have to give the reason in 2 to 3 sentences and you should mark the keywords. Even though ನಿಮಗೆ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಆಗಿದ್ರು ಸಮಟೈಮ್ಸ್ ಏನಾಗತ್ತೆ ಅಂದ್ರೆ ಕಾಮನ್ ವರ್ಡ್ಸ್ ಅಲ್ಲಿ ನೀವೇನ್ ಮಾಡ್ತೀರಾ, ಯು ವಿಲ್ ಟ್ರೈ ಟು ಎಕ್ಸ್ಪ್ಲೇನ್. ಸೊ ಸೊ ಆವಾಗ ಕೀವರ್ಡ್ಸ್ ಬರ್ದೇ ಹೋದ್ರೆ ದ ಇವ್ಯಾಲ್ಯೂಯೇಟರ್ ಕೆನಾಟ್ ಗಿವ್ ಯು ದ ಮಾರ್ಕ್ಸ್. ಬಿಕಾಸ್ ಯಾವ ರೀತಿ ಮಾರ್ಕ್ ಸ್ಕೀಮ್ ರೆಡಿ ಆಗಿರತ್ತೆ ಅಂದ್ರೆ ಇಷ್ಟು ಕೀವರ್ಡ್ಸ್ ಇರಬೇಕು ಅಂತ ಇರತ್ತೆ ಆನ್ಸರ್ ಅಲ್ಲಿ. ಓಕೆನಾ? ಸೊ ದೋಸ್ ಕೀವರ್ಡ್ಸ್ ಆರ್ ವೆರಿ ನೆಸೆಸರಿ ಹಿಯರ್. ನೆಕ್ಸ್ಟ್, third question. What is polymerase chain reaction? Name the bacterium from which the polymerase enzyme used in this technique is obtained. Write the schematic presentation of this technique. See, this is one question, 3 sub questions ide idralli. Polymerase chain reaction andre enu? That is 2 marks. Okay. Name the bacterium from which polymerase used in this technique. That is 1 mark. Write the schematic presentation. Okay, that is 3 plus 1. Sorry, 2 plus 1, 3, 3 plus 2. 2 marks. ಹೌದಾ? ಸೊ 2, 1, 2 ಆಗಿ ಈ ಕ್ವಶನ್ಸ್ಗೆ ಆನ್ಸರ್ ಏನ್ ಮಾಡುತ್ತೆ? ಡಿವೈಡ್ ಆಗ್ತವೆ. ಸೊ ವಾಟ್ ಈಸ್ ಪಾಲಿಮರೇಸ್ ರಿಯಾಕ್ಷನ್? ಜಸ್ಟ್ ರೀಕಾಲ್. ಏನು ಪಾಲಿಮರೇಸ್ ಚೈನ್ ರಿಯಾಕ್ಷನ್ ಅಂದ್ರೆ ಯಾಕೆ ಮಾಡಬೇಕು? ಟು ಗೆಟ್ ದ ಮಲ್ಟಿಪಲ್ ಕಾಪೀಸ್. ಸೊ ನಮ್ಮ ಜೀನ್ ಆಫ್ ಇಂಟ್ರೆಸ್ಟ್ ಸ್ವಲ್ಪ ಇದ್ರೆ ನಮಗೆ ಎಕ್ಸ್ಪೆರಿಮೆಂಟಿಗೆ ಸಾಕಾಗೋದಿಲ್ಲ. ಸೊ ಅದರ ಕಾಪೀಸ್ಗಳು ಬೇಕಾಗ್ತವೆ, ವಿಚ್ ವಿ ವಿಲ್ ಸಿ ಆರ್ ವಿ ವಿಲ್ ಗೆಟ್ ಇನ್ ಪಾಲಿಮರೇಸ್ ಚೈನ್ ರಿಯಾಕ್ಷನ್. ಸೊ ಲೆಟ್ us see how to write the answer. Yeah, see here, polymerase chain reaction. So in this reaction, multiple copies of the recombinant DNA, or what we can say gene of interest, is synthesized in vitro inside the laboratory using 2 sets of primer. Primer andre small chemically synthesized oligonucleotides, and the enzyme DNA polymerase. So DNA polymerase, DNA primers ಅನ್ನ ಯೂಸ್ ಮಾಡಿ ನಾವೇನ್ ಮಾಡ್ತೇವೆ? ಮಲ್ಟಿಪಲ್ ಕಾಪೀಸ್ ಆಫ್ ಜೀನ್ ಆಫ್ ಇಂಟ್ರೆಸ್ಟ್ ಅನ್ನ PCR ಮೂಲಕ ತಯಾರು ಮಾಡ್ತೇವೆ. ಸೊ ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದ ಪಾಲಿಮರೇಸ್ ಚೈನ್ ರಿಯಾಕ್ಷನ್. ಸೊ ವಿಚ್ ಆರ್ ದ ಕೀವರ್ಡ್ಸ್ ಹಿಯರ್? ಮಲ್ಟಿಪಲ್ ಕಾಪೀಸ್, ಇನ್ ವಿಟ್ರೋ, set of primers, right? DNA polymerase enzyme. Next we have is, next they asked the question, from which bacteria they will derive the enzyme which is used for polymerase chain reaction. So ಯಾವ enzyme ಬೇಕು ನಮಗೆ ಇಲ್ಲಿ? So we want DNA polymerase enzyme. So this enzyme is usually obtained from a bacterium which is called Thermus aquaticus anta. From this bacteria they will get the polymerase enzyme. Then what is the question? They asked us the schematic representation. See here. So here you can see, let me put the laser. Yeah. So schematic diagram andre you should label it also. So idu DNA, double stranded DNA. So adralli which is colored alva. So that has to be amplified. So the first step is denaturation. You are doing it by heating, where the separation of fragments will be done. Next, annealing, where you are introducing the primers, right? Then DNA polymerase enzyme will take the action. Next is extension. So the extension, that is complementary base pairing, will occur. 30 cycle centrifugation will be done, and from 1 copy you got 2 copies, right? So 30 cycles, right? So 1 billion time amplify agatte idu. So this is how the schematic diagram has to be done. So be aware while writing the schematic diagram, you have to shade the region which is gene of interest. You have to put 5 prime to 3 prime, 3 prime, 5 prime is necessary. Denaturation, annealing, extension methods has to be mentioned, and heat, polymerase enzyme action has to be mentioned, right? So if you draw like this systematically, then you will get full marks, what we say, right? So this is about the third question, I guess, right? Yeah. The next question goes like this. Again the same kind of question. Give reason for any 5 of the following statement. The first one is, origin of replication in a vector is an essential site required for gene cloning. Then selectable marker in a vector is essential site without which identifying rDNA becomes difficult. Next, vectors based need to have very few or preferably single recognition site. Selection of recombinant due to inactivation of antibiotic is a cumbersome procedure. Chilled ethanol is used in DNA isolation. So for every statement, you should give the reason. So let us check the answer here. What is the first question? Okay. So ಒರೈ ಸೈಟ್ ಅಲ್ಲಿ ಯಾಕೆ ರೆಪ್ಲಿಕೇಶನ್ ಆಗತ್ತೆ? ಸೊ ರೆಪ್ಲಿಕೇಶನ್ ದ ಒರೈ ಅಂತ ಬಂದಾಗ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಅಥವಾ ಪ್ಲಾಸ್ಮಿಡ್ ಅಲ್ಲಿ ಇಟ್ ಇಸ್ ದ ಸೀಕ್ವೆನ್ಸ್ ವೇರ್ ರೆಪ್ಲಿಕೇಶನ್ ಬಿಗಿನ್ಸ್. ಅಲ್ಲಿ ಏನಾಗತ್ತೆ ರೆಪ್ಲಿಕೇಶನ್ ಸ್ಟಾರ್ಟ್ ಆಗತ್ತೆ. Any DNA sequence linked to the ori will be replicated, right? Then when you see the copy number anta irutte, the ori controls the number of copies of linked DNA. Then cloning, a vector in a DNA molecule that carries a foreign DNA fragment into a host cell for cloning. The ori is a common feature. One vector ide antandre ori annodu adral irbeku, so that is a common feature. ನೆಕ್ಸ್ಟ್ ಏನಿದೆ ಸೆಲೆಕ್ಟಿಬಲ್ ಮಾರ್ಕರ್ಸ್ ಬಗ್ಗೆ ಇದೆ. ಸೊ ಸೆಲೆಕ್ಟಿಬಲ್ ಮಾರ್ಕರ್ಸ್ ವೈ ದೇ ಆರ್ ಎಸೆನ್ಷಿಯಲ್ ಅಂತ. ಯಾಕೆ ಅಂದ್ರೆ ಆಸ್ ಯು ಕ್ಯಾನ್ ಸಿ ಹಿಯರ್ ಇಟ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ಐಡೆಂಟಿಫೈಯಿಂಗ್ ಅಂಡ್ ಎಲಿಮಿನೇಟಿಂಗ್ ನಾನ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್. ಸೊ ಈಗ ರೀಕಾಂಬಿನೆಂಟ್ DNA ಮಾಡುವಾಗ ಇಟ್ ಈಸ್ ನಾಟ್ ದಟ್ ಆಲ್ ದ ಪ್ಲಾಸ್ಮಿಡ್ ವಾಟ್ ವಿ ಟುಕ್, so it underwent the procedure and all the plasmids got recombinant DNA. All the bacteria got recombinant DNA. There are certain bacteria where this recombinant DNA will not be formed. So non-transformants ಯಾವ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ದಲ್ಲಿ recombinant DNA ಫಾರ್ಮ್ ಆಗಿಲ್ವೋ ಸೊ ಅದನ್ನ ನಾವೇನ್ ಮಾಡಬೇಕು, ಅದರಿಂದ ತೆಗೆದು ಹಾಕಬೇಕು, ಕಲ್ಚರ್ ಇಂದ ತೆಗಿಬೇಕು ಅಲ್ವಾ. ಸೊ ಬಿಕಾಸ್ ಇಟ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ಐಡೆಂಟಿಫೈಯಿಂಗ್ and eliminating non-transformants which bacteria which are not transformed and selectively permitting the growth of transformant. So that is why selectable markers are necessary. Next, what is the question? Yeah. So vector gave why vector has usually single recognition site anta. Yake? So presence of more than one recognition site within the vector. Vector alli ಒಂದಕ್ಕಿಂತ ಹೆಚ್ಚು ರೆಕಗ್ನಿಷನ್ ಸೈಟ್ ಇದ್ದಾಗ ಇಟ್ ವಿಲ್ ಜನರೇಟ್ ಸೆವರಲ್ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್. ಸೊ ಯಾವಾಗ ಸೆವರಲ್ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ಗಳು ಜನರೇಟ್ ಆಗ್ತಾವೋ ಇಟ್ ವಿಲ್ ಕಾಂಪ್ಲಿಕೇಟ್ ದ ಜೀನ್ ಕ್ಲೋನಿಂಗ್ ಪ್ರೊಸೆಸ್. ಸೊ ದಟ್ ಈಸ್ ವೈ ಇನ್ ಆರ್ಡರ್ ಟು ಲಿಂಕ್ ದ ಫಾರಿನ್ DNA or alien DNA, the vector needs to have a single recognition site where that foreign DNA will bind, so that the gene cloning will be easier. ತುಂಬಾ ಕಡೆ ಇದ್ದಾಗ ತುಂಬಾ ಕಾಪೀಸ್ಗಳು ಅಟ್ಯಾಚ್ ಆಗ್ತವೆ and it will be tough for us for mapping or gene cloning what we say. So vector will be having single recognition site. Next, inactivation of antibiotics. So cumbersome procedure ಅಥವಾ tough procedure ಅಂತ. ಯಾಕೆ? So selection of recombinant due to inactivation of antibiotics is a cumbersome procedure. ಯಾಕೆ ಅಂದ್ರೆ it requires simultaneous plating. 2 plates ಅಲ್ಲಿ simultaneous ಆಗಿ ನೀವೇನ್ ಮಾಡಬೇಕು, ಪ್ಲೇಟಿಂಗ್ ಅನ್ನ ಅಂದ್ರೆ ಆ ಕಲ್ಚರ್ ಅನ್ನ ಪ್ಲೇಟಿಂಗ್ ಮಾಡ್ತಾ ಹೋಗ್ಬೇಕು with having the different antibiotics. So that is why it is a cumbersome procedure. Next, chilled ethanol. Chilled ethanol ಅನ್ನ ನಾವು ಯೂಸ್ ಮಾಡ್ತೇವೆ. ಯಾಕೆ? During the process of isolation of DNA, we are using chilled ethanol. Why? Because it is known as, it has a low electric or a dielectric constant. Water kinta adralli dielectric constant. You will be aware of dielectric constant in chemistry. So adara dielectric constant enide kadme ide. So adu dielectric constant yavag kadme iratto, it promotes the ionic bond formation. So it promotes ionic bond formation, making the DNA to precipitate. Fragments ಅಥವಾ thread like structure ತರ ನಮ್ಮ DNA ಏನಾಗುತ್ತೆ, precipitate ಆಗತ್ತೆ with the help of this ethanol, right? So this is the question. Next one is, we have a diagrammatic question. Explain structure of PBr322 with a neat labeled diagram they asked. Sometimes ಏನಾಗತ್ತೆ, so 5 marks question usually ನೀವು at the end of the exam you will attend this question. So due to the lack of time or in a hurry or in an anxious way, what you do, you just draw the diagram and label it and leave it. But what they asked in the question, explain the structure of PBR322 with neat label diagram. You have to explain it. Then you have to draw the neat label diagram. So how to write it? Try to write like this if the page is good. Okay, if the page is enough. Or write the first explanation. Below you write the diagram. You draw the diagram. So, ಏನಿದು PBR22 andre? We know that it is a plasmid. P stands for what? Plasmid. BR stands for what? Bolivar and Rodriguez, who discovered this plasmid, right? So, they have 4,361 base pairs. So, this plasmid has 4361 BP, that is base pairs. They have two antibiotic resistance gene, as you can see in the diagram. So, ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, whatever you explain in the explanation, ಅದು ನಿಮ್ಮ diagram ಮೂಲಕ ಏನಾಗ್ತಿರ್ಬೇಕು? It has to reflect. See, ampicillin resistance gene ಇಲ್ಲಿ ಕಾಣಿಸ್ತಾ ಇದೆ ಅಲ್ವಾ? Tetracycline resistance gene ಇಲ್ಲಿ ಕಾಣಿಸ್ತಾ ಇದೆ. ಹೌದಾ? It has the origin of replication sites, ori. It has more than 40 restriction sites ಗಳು ಇವೆ. Okay. There are 11 sites in the tetracycline. ಇದರಲ್ಲಿ 11 sites ಗಳಿವೆ. ಓಕೆನಾ? And 6 main restriction sites in ampicillin. The gene of resistance can be amplified using chloramphenicol. It is a double stranded molecule. E. coli illide hind 3 BAMH2. So here somewhere it will come, are some of the restriction enzymes on PBR322. Alright, so this is regarding what? The PBR322. Right, you have to draw this diagram and you have to explain it. At least 5 to 6 points you explain. 3 marks will be for explanation, 2 marks will be for the diagram. Okay, now? Yeah. So here, while drawing any diagram, below it, just write the word, the diagram represents. So the sentence should be like this. Below the diagram, okay, as I don't have space, let me try to write below. Yeah, so below every diagram you write the diagram represents. So this diagram represents what? Represents E. coli PBR22, E. coli PBR322. Okay, so if you want showing restriction sites and restriction genes, if you remember, you can write this or no. It is E. coli, it is not E. coil, it is E. coli. Yeah, so this is how you have to draw every diagram, not only in this chapter. If you draw the diagram of what we can say, the schematic diagram, what we saw regarding PCR. So below that diagram, you have to say this schematic diagram represents polymerase chain reaction and the steps involved it. That is how you will represent the answer. It is not that only in a PU paper you have to write it. So if you attempt any competitive exam in your future, like UPSC, KPSC, right? So in that also, this is a way to write the answer or to draw the diagram and represent it. Fine. So let us proceed to the next question. Yeah. Yeah. So the next question goes like this. Describe the characteristics that a plasmid should possess. One second. Should possess to be used as a cloning vector in genetic engineering. So if we have to use any plasmid in a genetic engineering, what are the characteristics it should have? Then, how is a bacterial cell made competent to take up the recombinant DNA from the medium? So here we have two questions, right? 3 plus 2. Let us see the answer, how we have to write it. First, a cloning vector must have the following characteristics. So write 3, what we can say, steps. It should have origin of replication or what we call ori. Selectable markers should be present, which helps us to identify the transformants or non-transformants. Then cloning site or recognition site for restriction enzyme to recognize, that should be present. So these are the 3 key characteristics which are needed for a cloning vector, which has to be used in the genetic engineering. Next we have is, what is the question? How is bacterial cell made competent to take up recombinant DNA from the medium? So what is made in the bacterial cell to take up the rDNA from the medium? What changes are made? DNA means what? It is a hydrophilic. Philic means what? Love towards water. Therefore, it cannot pass through the cell membrane. The bacterial cells, what we selected, it can be made competent. How? By treating with a specific concentration of divalent. What do you mean by divalent? 2 minus 2 plus 2. That is a divalent, right? Calcium 2 plus. The cells are then incubated or ice followed by a heat shock by placing them briefly at 42 degrees Celsius temperature and then putting back in ice. So ಇದರಿಂದ ಏನಾಗ್ತವೆ ಅಂದ್ರೆ the bacterial cell becomes competent to take up the recombinant DNA. So these are about what we say certain 5 marks question which we have to focus on. Right. The next question which we have is explain briefly the steps involved in recombinant DNA technology. So they asked explain briefly. Sometimes what happens? Certain students will not remember the explanation. So the certain students who usually by heart, no. So they will not remember the explanation. They will remember the steps only. So that time if you write steps only, at least if you write the steps, you will get 2 marks. If you write the steps with the explanation, you will get full marks. So let us see how to write. So this is the way I just showed you how to write it in a what we can say a pictorial way, pictorial way. So this we saw in our previous session also. So process of DNA technology has step 1 where isolation of DNA. So the subheading should be isolation of DNA. So DNA with desired characteristic is isolated from an organism. Next we have step 2. The desired DNA is integrated with the DNA of a vector. This is done as DNA fragment needs to be replicated. This is the second step. The next one is it is linked with the origin of replication of a vector. Thus it is multiply itself in the host. Replication subheading. Next, the newly formed DNA is a recombinant DNA. Next, this DNA is insertion into an organism to incorporate the desired characteristics, or the vector is used for the production of genetically modified products. So this is how you have to explain. So this one answer I just did not write with the words. So I just showed you how pictorially also you can write the answer. So no doubt it takes you time to draw this all. So you can use any other method. You can just put the box one by other and you can just explain the steps, right? So or steps you can use 1, 2, 3, 4 like this, right? If you have enough time. Okay, so that is also a way of representing the answer. Fine. Yeah. See here, this is a pictorial way, recombinant DNA technology. So what happens here? Agrobacterium tumefaciens treat foreign DNA and plasmid with restriction enzyme and DNA ligase, right? So from this we will take a T plasmid agrobacterium. DNA containing gene of interest will be selected and it has to be inserted here. So this is recombinant T plasmid. The second one is what? Introduce the recombinant plasmid into cultured plant cell. Inserted DNA carrying new gene. The next one is regenerate the plant from the new cell. So this is how the diagram of the recombinant DNA looks. This also you can use. Next, yeah. So these are the questions. Yeah, these are the 5 marks questions related to biotechnology principle and processes. Fine. So we have solved around 7 questions which carries 5 marks. So in this, they may ask the high order thinking question like give reasons, or will explain with a schematic diagram, or they may ask a direct question also. But according to the new blueprint, what we see, according to the new blueprint, so the whatever you get the 5 marks question from this chapter, it will be of high order thinking question. Okay, so prepare according to that view. Okay, so these are the most important question which I discussed. If you know any other questions also, you can find the answer. If you don't find the answer, please do comment the question. I will be there to answer you. Okay, fine then. Let me end up the session here. Have a nice day. Take care. Bye-bye.